Good afternoon. I'm Gula Lipschitz, a biotech analyst at Chardon, and over the past year we've seen the landmark first approval of a CRISPR nuclease therapy for sickle cell and beta thalassemia. We've also seen initial in vivo nuclease knockdown programs enter pivotal trials and in vivo data on base editing. The more mature programs in vivo are focusing on gene silencing, but we're also seeing scientific progress on gene insertion and gene correction that could unlock additional applications and expand the commercial potential of genome editing. So in this session, we'll talk about the scientific, strategic, and commercial considerations for these technologies in the context of that evolving space. So I'm joined by Dr. Keith Gottesdiener, CEO of Prime Medicine, and Dr. Brian Thomas, CEO of Metagenomi. So thank you both for joining us. And just to jump in my little intro, I just mentioned that the more mature in vivo clinical stage programs from Intellia and Verve have focused on using the tech for knockdown, aiming to provide a deeper target reduction or better convenience or compliance through a one-time dosing versus the available or emerging RNAi approaches and the like. Gene insertion and correction approaches are generally going after a different set of indications that, for example, allow you to restore a non-functional gene. So can you talk a little bit about what the attributes of indications that can be addressed by gene insertion and gene correction that are attractive from both a scientific and a commercial standpoint? So we can pick one to go first. Brian, go ahead.
I'm happy to start. I think from a technical point of view, it's really exciting to see. I think it's funny that we're talking about the first wave of these companies that are using this revolutionary technology of programmable nucleases to knock genes out for therapeutic curative treatments, and now sort of just pushing into what's coming next. And I think it's interesting that there's a demand that we know gene editing technology is going to make a difference. We know that it's coming. We know that it's going to be powerful. And I think one of the reasons that you saw so many people focus on the liver and focus on specific indications is because that particular modality, that's what it does. That's what it's capable of doing. And it's really taken the evolution of these new capabilities like base editing, like RNA-mediated integration or prime editing, larger type integrations with transposase and directed transposases to really get us to start to think about other ways that we can interact with the human genome. At Metagenomi, we've really made that a core tenet of what we've tried to pursue, and our strategy has always been to not be limited by the tool and be able to think about the genetic disease and really be able to say, look at across the landscape of genetic disease and be able to say, how are we going to treat that disease, and what does the tool look like in order to be able to do that? And I love the title of this panel because it's really focused on that later stage. As you think about complex genetic disease, it becomes diseases like cystic fibrosis, where you don't just have a single correction that you need to make in order to cure that disease. The population brings thousands of different possibilities. And so, in order to correct those with the sort of, again, first gen of programmable nucleases, or, you can't, right? You have to think about other ways to do it. And so for us, we've really tried to focus on making sure we have that flexibility of when we approach genetic disease, we're bringing the right genetic modality.
Mm-hmm. So I agree obviously with much of what Brian is saying. I think that if you knock down a gene, which is really what many of the early work has been done, sorry, a lot of noise in the background. If you're knocking down a gene such as that, there are wonderful places to work. But when you look, there's a dearth, comparatively speaking, of places where that technology can work. I think when you begin to talk about correcting a gene, which is really what Brian and I are talking about, different techniques in many cases, but really correcting a gene, you really have to reach deeper into your toolbox to be able to do so. And I think it just opens up new possibilities. Once again, there's a gradation of things, and I don't mean to suggest it's a gradation of value, so that things at the bottom of the gradation are less valuable and things at the top are not. But you could think about things that are closer to gene replacement in some way, to things that might be truly a correction where you can go in and you can bring things back exactly to wild type, to normal. And when you're done, that genome looks exactly the way it might look in someone who didn't have that disease. Wherever on that gradation you want to go, you have to ask yourself, what is the tool that one needs that can provide, where one gets to really make a difference overall. At Prime Editing and with Prime Medicine, we are very attached to the idea that making those changes in the endogenous locus with a corrective mutation in some ways just kind of gets us excited, is the only way to say it, because when we're done, it is an example, hard to do, but an example of where when you're done, that gene behaves exactly the way it would have as if a mutation didn't exist before. And we think that's going to be something that's very, very interesting. Is that necessary in every disease? Absolutely not. And once again, it's a matter of picking the tools that one wants to use and figuring out the best way to use them. You can really make extraordinary changes. And I got to tell you, Gula, you talked about it. This is now. I mean, when I look ahead a couple of years, I think we'll be doing this pretty regularly in the clinical trials. You go out 5 years, and I think we're going to see lots and lots of examples where genetic editing is well on the way to a cure for a disease. I mean, the energy in this area is just extraordinary.
And I think further to that, these first generation technologies have done something really important for the gene editing ecosystem, which is they've blazed a trail with the regulatory agencies, and I think that that's also important. You have to think about, we're talking about changing the human genome as a treatment. That is, it's a new paradigm that the world is kind of coming up to, and so I think it's important to keep it, the whole ecosystem in mind as we think about genetic editing and where it can take us.
As we think about the type of indications that you can go after with this type of approach, obviously some of these have been explored using viral or non-viral gene delivery. Of course, one-time dosing offers convenience if the existing approach has been non-viral or enzyme replacement or something. But more generally, are there advantages that you see to using gene correction or gene insertion for some of these applications that have been explored with other modalities?
So I definitely think so. Listen, I do smile a little bit. I worked for years at Merck, and I remember when Merck looked at the first agent, for example, in diabetes that required an injection as a result of that. It took you got a week's worth of coverage before you had to treat that patient again. And Merck said basically, no one's going to care about the fact that you only have to treat once a week instead of once a day. Well, that's the GLP-1s that developed. And then within the GLP-1 group, it became, do I have to treat once a week, once every 2 weeks, once every 4 weeks? People are built companies around once yearly. The same thing is true when one looks at other modalities, like some of the approaches to knockdowns. So in many ways, the end result of that is once and done. So I do believe that many patients are going to care about, can I get a procedure, whatever it is, procedure is probably not the right word, a treatment, that in the end, when I'm done with it, I truly am cured. I think that's going to appeal to many patients as they go forward, that this disease is over. It's not that I have to come in every month or every 3 months or every 6 months to get a treatment. Frankly, I just never have to worry about it again. I think that's going to be a major value to patients as you go forward.
Keith, you bring up a really good point around the sort of mental framework that people suffering from these diseases have to deal with. And for our lead program in Heme, we have a fantastic SAB member who's a luminary and also a Heme patient himself, who talks about this hemophilia brain, where patients are constantly concerned about their lifestyle and their habits because they suffer from this. And I think that it's a real opportunity for gene editing to shine as an application. And then I think specifically, I'm not sure if you wanted to talk about the viral, non-viral aspects and how these new technologies kind of fit there, but I think that's also really important as well. And that's one of the core tenets of why we decided to build a diverse portfolio of gene editing capabilities, is to really be able to think about not only the the common targets that you can get after in the liver with knockdowns or other types of gene editing corrections, but also being able to get outside the liver. And so, in the case of programmable nucleases and others, especially when you add a programmable nuclease plus a deaminase for a base editor or plus a reverse transcriptase of some type for other types, you really start to get a limitation on where you can go with viral type deliveries. And so one of the areas that we've actually been able to leverage our discovery platform is in finding very novel programmable nucleases that are a fraction of the size. And so it's actually allowed us to perform the experiment of a very small size programmable nuclease with small size deaminases and reverse transcriptases that could potentially be packaged in an AAV. And again, really getting this out into disease space areas that are not the kind of common targets of today.
So Keith, maybe I'll direct a question to you, and then I have one for Brian. So for Keith, what do you see as the biggest challenge today with respect to implementing precise mutation correction?
I don't actually think there are challenges the way you phrased the question in that way. We believe we can do that today in many diseases. If you ask the question of if you wanted to really deliver on the whole promise of what those kinds of changes could do, probably the biggest limitation today, I would say, is still delivery, because our ability to get at the right level in all of the tissues we want to get to is somewhat limited. Now, keep in mind, that's an amazing thing, okay? That's the problem. Sorry, not that delivery is a problem, that's not amazing, but the fact that delivery turns out to be that amazing problem. And the reason I say that is I had a whole list of those problems that were really major problems 4.5 years ago when I joined Prime Editing. Could we really do this safely? Check. Can we actually do the complexity of edits? Check. Can we develop a toolbox of things that we could use? Check. Check meaning, of course, these are things that are well on the way to being solved. So along the way, we've knocked off problem after problem. The delivery problem isn't solved. But I will tell you, I tell this story often, so I apologize if you've heard it before, but at that point, back in 2020, when I went looking for delivery companies, because when Prime started, our philosophy was, we're never going to do delivery, we're a gene editing company. We're the payload, we'll find a delivery company and we'll pair with them, we'll split the profits some way, and we just are never going to build that in. You went looking, it was almost impossible to really find a delivery problem then. Look at it today. Today, delivery companies are everywhere. In some ways, that's part of the problem. Everybody owns a piece of the technology. It's very hard for us to figure out who to talk to and such. But there's so much enthusiasm, so much good work going on. I'm just incredibly hopeful that over the next couple of years, our ability to get to organ after organ after organ, it's just going to get better and better every day. And in fact, there are places, once again, a couple of years ago, if you had said to me, would someone seriously say that, yes, I think I could get to the brain at a therapeutic level, I would have said, what are you smoking, okay? But today, when companies make that claim, I think many of them are well on their way to getting there. I'm not sure all of them will get there, but it's a reality again versus where it was, a couple of years ago. I'm very enthused by that.
Brian, do you have any thoughts on this front?
I totally agree with the enthusiasm behind the innovation that we're seeing in the delivery space, both from LNP as well as viral and other creative means. And I think it's very exciting where we can go. And I think we're also accentuating the challenge. We know that this is revolutionary technology and it's going to be able to make an impact on humanity, but delivery has to be a critical element that evolves along with us.
And maybe to direct to Brian first.
Sure.
So, you guys are advancing a bunch of different technologies that could do whole gene insertion or gene correction, whether that's nuclease-based, RT-based, transposase. Can you talk a little bit about how you're thinking about these different technologies with respect to each other and how you choose what to deploy for a given indication?
Sure. So that kind of comes back to the way that the company was founded. And we set up a strategy early on that we have an ability to go into the natural environment and find novel enzymes. And when we started to do this, we had my academic co-founder and I, Jill Banfield, we had a paper with Jennifer Doudna showing that you can leverage these metagenomic data sets to find alternatives to Cas9 that have different characteristics. We had no idea that it was going to be as extensive as what we ended up seeing. And so we now literally have thousands of these systems, probably more than 20 that we have leveraged as chassis within the organization, and also, as you mentioned, are driving both our knockout programs with our partner Ionis, but also our lead program where we're using it to perform a large gene integration. But it was a strategy that we set up then, which is, we know that there are other capabilities that we can build on with this chassis, but we also have these amazing nucleases that are working right now, and they're effective, and they're efficient. They show no off-target activity. They're very small. We can think about where we can send them outside of the liver. So that, to us, that's something tangible we needed to grab then. And so our strategy was move forward with technology that's ready now, coupled with delivery that's ready now, and try to make progress there. And so that has kind of been what's driving us. But now I think it comes back to the first question for us. As we have developed these other capabilities, including our Cas system, our CRISPR-associated transposase system, that theoretically has the ability to deliver tens of thousands of KB in a specific location. Now we can start to think about diseases that are much more complex than what our original strategy was when we set out.
Got it. I'm going to take a hard veer to a different topic, which is stock performance and how people are valuing genome editing companies. Obviously, as you just talked about, there's been exciting progress on the science, but from a stock perspective, several of the gene editing companies have been having a choppy time on the market, so to speak. So can you guys talk a little bit about why you think that is and what could change that?
So maybe I'll go first on that one. So it's been a terrible time for gene editing companies, and people are looking at us as a group, and really very little differentiation between those gene editing companies or any kind of related technology. Now, of course, I hate that because, of course, being a CEO,
I hate it too.
I personally believe that we have the best, and it's time for people to wake up and see just how different we are. All kidding aside, though I do actually believe that, but all kidding aside, because there's many great technologies out there, I don't think people really know how to value us, and I think people are starting to realize something that everyone in the companies knew, which is when you start to develop a brand new technology, it isn't as if you can snap your fingers and everything just suddenly falls into place. The story that we tell, not we, but the community tells, is pretty extraordinary. But I'll just tell you the way I look at it from the prime editing point of view. 4 years ago, prime editing was done in one laboratory anywhere in the world, okay? It was literally just being published. 4 years later, we're in the clinic with prime editing technology, and we have a variety of programs going forward. By any stretch of the imagination, that's an extraordinary path. There are a few examples that are faster, not many. So I do think that, people don't really understand it, and that caught up to us a little bit, and I think that'll smooth itself out as these catalysts, these delivery, these data come out over the next year or two. I really foresee the next couple of years as amazing things will be delivered. Investors will see this is real, and they'll begin to look at it a little bit different. Meanwhile, it's terribly frustrating, and any of you who would like to particularly help either of our stocks along, we'd be very excited if you decided to make a major investment today.
Yeah, I agree. It's obviously been a really challenging time, especially we just went public this year, and it's been extremely challenging. As the founder of the company, I feel like this company's my baby, and I have 2 decades of academic experience with pushing this technology to this point, and I know it has value. I'm excited about this company. I'm excited about the ecosystem. I think you hear that from both Keith and I. I think what I am trying to show the world is we set out a strategy, And we are going to show you systematic execution towards that strategy. And we are translating the technology one step at a time. But as Keith mentioned, this is going to take time. This is a brand new field. And it's revolutionary for a reason. It's just going to take time to articulate what that looks like. And so I echo what Keith just said. I try to get people excited and tell them about the science and the potential and where it can go and tell them about our milestones. But I can't magically pull out a phase 3 in the next quarter to show people that I'm in the.
Can I just say one other thought that I have? I mean, when Prime was settled, there was no relationship with milestones and valuation at all. That was probably good for us in many ways, but essentially, to be fair, at that point there was more hype than reality. I don't mean we were doing the hype, but hype in terms of people's expectations, so that those two are very unrelated. Today, I feel exactly the same thing is true. There's no relationship on what you're delivering and the value as investors look at it. Either way you look at it, something is really wrong, and I think it will correct itself. What we did is we went from one extreme to the other extreme in a very rapid way. If we get to the point someday where what the companies are delivering and the value that they receive begin to come back in sync, I think many of these companies are going to do very, very well for themselves, because what we've been doing during that period of time when there's that dissociation, that lack of correlation, frankly, it's just been delivering, executing. And eventually, I personally believe that execution will reflect in value. From my point of view, Gula, this is speaking seriously, not really jocularly as I did before. I think these companies have extreme value, and at these prices, they should be very, very attractive. If you have a viewpoint that can go out, let's say 2 years, I think everybody's going to be in a good position who invests in these companies. But today, it's a little bit harder to know exactly what to do, because I don't think there's anything any gene company has done in the last 6 months that's been reflected in a change in their stock price. It's just, it's disheartening, but it is the way things are.
I agree, and I think that the nice thing is, though, like to your conference today, Gula, we've had a full slate of investor meetings, and the investors are still engaged and still interested in the technology and in the development of the space, and for that, I'm really excited. And also, I think we're seeing a lot of interest from potential BD partners, and you see today some actual BD that's happening in the space. To me, I think that's a very good sign and a very hopeful sign.
And I would agree. I mean, to make a shameless plug, we did a beautiful deal today for those people who don't know. It's a major deal with BMS. I'm not going to say much about the actual deal here. That wasn't my point. My point really is it shows that pharma is interested in investing in this area, and eventually that'll sink into other deals and also help people to understand why there are opportunities here. I don't think it's the only reason these companies should be valued, but it certainly is one very important ancillary point that really, is just a little bit different today than it was yesterday. It's been a long time since someone's done a 9-figure deal up front, for a gene editing type of approach. And I think it's been long overdue. I'm pleased it was us, but frankly, I would have been happy, not as happy, but I would have been happy if any company had actually achieved that, around this time.
Congratulations.
Absolutely. Congrats. Oh, good timing. I think we're just at the end of the session, so I'd like to thank both of you for the great discussion.
Well, thank you.
Thank you.